Get away.
All right, good evening, everybody. My name is Lisa Gonzalez, as Jerry said. I am currently in the entomology department collection area. And it's going to be a little tricky because the space that I'm in, it's kind of actually like a narrow office space, but I'm going to do my best to do sort of a combination of a demo with the digital microscope that I have the great privilege of being able to use for the BioScan project. So I'll do a little demo and then I'm going to show you some of my photos. And actually, I'm going to do that in reverse. I'm going to show you my photos and then I'll do a demo. But right behind me, sorry for the squeaky cart, the laptop is on a very squeaky cart. This is the incredible digital microscope that I get to use. It's called a Kienz. This one is the 5000 model. We've had this for, I believe, about 4 years now. This is something that was purchased by all of the RNC department here. So it's not just specifically for the entomology department, although it is housed in the entomology collection area. So when I get to work, sometimes there are paleontologists in here taking photos of things in amber. Our ornithologist was in here taking photos of feathers the other day. So it's really exciting for us because we get to kind of, keep track of what other people are doing and actually get to see it in action, I should say. So let me go ahead and now that you've kind of got a little, got to see what it looks like, let me go ahead and share my screen here. And give me a second. Here we go. It's opening up. How's that look? Can everyone see that? Maybe like a thumbs up if you can. Okay, great. Thank you so much. And then let me go ahead and go to the, no, let me not do that. I promise I've done a presentation before. Here we go. Here we go. All right. So just a little bit of context. I know Jerry mentioned the BioScan project, and I think a lot of you that are here tonight know what that is. But in case this something that you haven't heard of yet, the BioScan project is an insect biodiversity survey, specifically in the Los Angeles area and some surrounding counties. So we have had some research sites that were going out into San Bernardino and Riverside. But what's unique about BioScan is that it's the first large-scale urban insect biodiversity project where we use malaise traps in backyards mostly, but also some community spaces like school gardens and community gardens as well. So, we had people who signed up. We've had about 80 different participants, both individuals and families, including some schools, that very graciously allowed us to put a malaise trap, which you could see is not a small trap. This is not like putting a little sticky trap in a citrus tree. I mean, this is a significantly sized trap in their backyard, letting us leave it there for at least a year. Some people had their trap up for multiple years, and then helped us out as community scientists by changing out the bottles and helping us to monitor the trap. And then on the left, you see kind of an example of sort of two extremes of what the samples look like after we leave them up for about a week. Those samples there were actually taken in our nature gardens right here at the Natural History Museum. And a pretty interesting contrast you could see before we finished planting and building and planting the nature garden. When it was under construction, you could see kind of how light that sample is. And then on the right, you could see what it looked like after everything was in full bloom and all of the various insects started to show up and really make the nature gardens their home. So when I get a sample, it basically looks like bug soup. And myself, along with a big team of people, this is not all me, we take those samples, we clean them up, we start to identify them down to the order level, and then with certain groups we're identifying down to the species level. Jim Hough, who's here tonight, is one of our collaborators that helps us to identify some of these insects. So my main responsibility, in addition to going out and monitoring the traps and being the liaison with all of these wonderful people who are helping us with the project, is to identify the insects. But in the process of doing that, I started to really wanted to do more outreach. I wanted people to really understand how amazing the insect diversity here is in LA, because usually things from the tropics or from faraway places is what gets all the attention, right? But we have amazing insect diversity in backyards, in an average backyard. We have incredible stuff. Well, most of those things are very, very tiny. So how are we going to take something that's super tiny and get people excited about it? If they're going to look at it and it looks like a speck of pepper, how are they going to, really understand the complexity and how amazing these things are? So I have just a couple of photos here just for scale. I think most of the people in this meeting know that most insects are really small, but for the general public, that's actually really surprising news. Often when you show them something, they're like, what? That's an insect? What? That's a living thing? They get really astounded by that. The photo on the right is photo. So that's actually a forward fly on the top, the head of a pin, an insect pin, just to show you how small it is. And that's actually one of the families that we mainly work on here. Our curator is a world expert on forward flies. So I spent a lot of time looking at teeny tiny flies and identifying them to the species level, which is for me is like a perfect day. All right, so here's just another photo. This is one that I took again to try to get people to understand this like sense of scale. The wasp on the top, that's a paper wasp, a real common wasp that people will see. And when I talk about wasps to the general public, this is what people usually think of. And they say, oh, I don't like them and blah, blah.
Hi, Honey.
Okay. Well, the vast majority,
I wanted to see what you're.
Somebody's not muted and someone just said, hi, honey. So I want to make sure if you're having you don't want us to hear this conversation. Okay. Or maybe I shouldn't have said anything. Anyhow, so the wasp on the top, that's a paper wasp. And then if you see the wasp that's underneath, that's circled, that's a trichogrammatid. I don't know what genus it is, but just to give you a sense of how small the majority of the wasps or the insects in general are that I'm looking at when I'm sorting through these samples. And they're just wondrous. And I wanted to really, I really wanted to share that with not just with all of our participants, not just with museum visitors, but with the world, really. So here are a couple examples. Jerry asked me to make sure to include photos that nobody has seen before. So I have some Lorquin exclusive photos here. A lot of these other photos I have shared in a variety of ways. I have a Instagram account that I haven't been so good at updating lately, but that's there. And then we also have an exhibit at the museum. So some of these photos are things that have been shown before. But for instance, this one on the left, this is this beautiful terra mallet. You can see all the sculpturing. I should have put the size there. I think this one is maybe about 4 millimeters, I want to say maybe. And this is one that was collected in Los Feliz in a backyard that's not too far away from Griffith Park. And then the one on the right, actually, this one's pretty exciting because a lot of these insects that I'm showing you, I'm telling you one location that it came from, but we see them in a lot of different samples across the LA basin. But the one on the right, that eucharitid, that's the only one that we've ever found. So this is not a particularly common wasp. And in fact, when I started working on this project, I knew of eucharidids because John Harity at UCR was one of my advisors. Actually, I worked in his lab for a while. And I knew about him because he studies eucharidids, and I thought they were all tropical. I didn't even know we had any in the LA basin. So when I saw this, I said, wait, is this really what I think it is? And I, automatically immediately sent it to him, and he was super excited that we found one in LA. So it was known to be here, I should say. But yeah, this is a really cool looking little wasp that's actually a parasitoid on carpenter ants. So it needs to be in areas where there are healthy populations of carpenter ants. So this one was actually right on the edge of the Verdugo Mountains. And yeah, we haven't seen any others in our traps other than this one individual. This is another photo I haven't posted anywhere yet, but another type of wasp that I should go back and say that before I started working on flies, when I was in school, I was mostly studying these small, what we call the Microhymenoptera, these parasitic wasps. And it's one of the groups that I'm the most enamored with, just because of their diversity and because I love their life history. Like when people ask me what my favorite insect is, I just say the parasitoids, the weird ones. Those are my favorites because their life histories are just so like something out of science fiction, but science, not fiction, right? But real. So this is another wasp, type of wasp, this family that I'd seen specimens of before. And I knew that we had them here in LA, but we started to get a handful of them in our traps. And I got so excited when I first started to see them turn up. So these are also, I didn't put the location there, but these are ones that we're finding along the periphery of Griffith Park. And they are parasitoids on beetle larvae living inside of wood. You can see that long ovipositor there, which they use to inject their eggs into their hosts. And those incredible little crowns that I understand are something that you see with some types of wasps that are going to pupate outside, or when they're pupating, they have to kind of bore out of the wood. So we believe that this is some modification that maybe somebody here knows more about that. But just an incredible little beastie there. Let me make sure I'm not talking too long here. Okay, I'll speed it up a little bit. And so then another thing, I mean, in addition to wanting to take things that are really tiny and blowing them up so that people can really see all of that complexity, I also really like to do what I think is like insect portraiture. And this is not always easy to do with a specimen. You know, when you're taking things that are living, you can kind of get a little bit more of the action of that animal. But as I'm looking through my microscope and as I'm looking at these organisms, there's the part, the scientific part where I'm looking at, you know, morphological features to identify it. But then there's also just the human in me that looks at them and just, you know, hearts come shooting out of my eyes. And I just think they're so beautiful. And this moment of an insect kind of looking at you is not something that we really get to experience, with the exception of maybe, you know, like praying mantises or something. But what I wanted to do with some of these photos was just really give people a sense of this animal looking at you. But I wanted it to be a positive emotional experience, not, you know, like this giant bug that's going to attack you, but just really, I really wanted for people to have some kind of emotional engagement with my photos. So I really like taking portraits. These are just a few examples. There's a syrphid on the left that has incredible stripes in its eyes. There's a beautiful bee on the right, a type of Anthophora. And then sometimes I'll do like more side angle portraits, or I'll, you know, like a straight, this is kind of a straight lateral, which is what we often do for publications. But again, I try to, and again, these are all specimens that they're not living. And so I'll sometimes try to position them in a way. So there's a lot that goes on before I take the photos. There's a lot of, you know, pinning them in a certain way, which is not always how we would pin them for a collection. So I'm only doing this with a handful of specimens just for this purpose. But it's always me kind of weighing like, okay, these are our research goals. This is what I need the specimens for. But occasionally I'll pull out a specimen and be like, you know, this is one that I'm going to use for a portrait. And so I'm going to do something a little different with it. Of course, the research is the main thing, right? So this beetle right here, this was the first time I'd seen one that turned up in one of our traps. And I think it's just, we don't tend to get a lot of beetles that are, you know, mostly leaf litter animals. Malaise traps are not what you use to collect them. But this little buddy climbed all the way up into the bottle, and it was just beautiful. It looks kind of more blue-green here, but when you see it in room lighting, it looks more purple. That structural coloration just kind of changes up a little bit when you put it under the scope. Okay, let me stop sharing, and then I will do just I'm seeing the comments now, the heart shooting out of my eyes. Yeah, exactly. Yes, totally understand. Okay, so I picked out a beetle here, a little click beetle that you can see. It has some nice fall colors. So that's why I picked it out for today. I already have it set up under the scope. What I do before I do that, I actually look at it under just another dissecting scope. I get a little brush that has just like a couple single little hairs on it. I try to take some of the dust off of it as carefully as possible. I mean, there's going to be editing after as well, but if I could get some of that dust, or, you know, what's on it a lot are the Lepidoptera scales, the worst. So I try to get some of those off and clean it up a little bit without, you know, doing any damage to the specimen, of course. I use a variety of different things. Sometimes I'll use a ring light. We have a couple of different types of diffusers. I'm actually using one of our lower lenses right now. So I'm going to take this at about like 100x, but we do have this is a fairly large insect compared to, you know, something like a 2 millimeter, 3 millimeter wasp. In that case, I would use one of our larger lenses with higher magnification. And then I also will use, where did I put it? Of course, I had it right here and then it disappeared. I can't find it quickly. Okay, here we go. So sometimes I'll use different sorts of things on the bottom just to kind of bounce the light around a little bit differently. This is actually when I take photos with a black background. I like to use this kind of shiny piece of acrylic, and it bounces the light a little bit differently. And this is all just from trial and error, me just trying different things. And also Brian, our curator, who also likes to do photography, he's also done some different things too, and I've kind of like picked up tips from him. So it's not all, you know, I don't want to give myself all the credit. So I'm going to place this under here very carefully. And right away you can see it like gives it better contrast, and that's just going to help me when I edit it. I mean, I could also just do this in editing too, but you want to do as much of it before and save, you know, you don't want to edit everything. You want to try to like make it the best photo you can first, and then so your editing only takes like 10-15 minutes. Ideally, sometimes it takes a lot longer. Okay, so I've got this little buddy set up, and then let me try to scoot this forward. I apologize for the squeaky wheels on this, but I just want you guys to be able to see what I'm doing here. And so I've got this lovely little controller here that allows me to adjust for depth of field. And this, I'm going to do 3D stitching right now. You can do just some, especially for something so flat, you could just do 2D stitching, just take care of your XY. But for this, I'm going to actually also make sure that we're adjusted on our Z axis as well. So I'm going to set up the range. I'm doing really quickly, okay, just for this demo. Just want to make sure that it's capturing the entire beetle here. I don't know if you guys can see that. Hopefully you can. I'm sorry, I know it's probably a little hard to see what's going on this side because of the lighting. And then I set the lower range. I raise up the lens, and then this is where you have to kind of make sure that you're capturing, because I picked this intentionally because there's one little antenna that's sticking straight up. And so you want to make sure that everything has been set in focus, because this is going to automatically stack. It's doing all this work for you. So, all right. And I think I did this right. I was a little distracted, so apologize if I did something wrong while I was talking, but let's see. Can you see that there? Yeah, okay. So you can see that it's making sure that it's raising up so that you're getting every kind of slice, and you want to make sure that you do with your depth of field that you get the bottom and the top clearly. So while that's going, because that might, this is going to take like a little less than a minute, not too long. If anybody wants to ask any questions, feel free. If not, I can tell you a little bit more about, I don't want to take up too much time because we have 2 other speakers.
And remember to take yourself off mute to ask the question.
What's the cost of that microscope?
Oh, yeah. So I was not part of the person who eventually purchased it, did some incredible negotiating on behalf of the institution. So I actually don't know what the final cost was, but these cost tens of thousands of dollars. They're pretty expensive. So this was paid for as part of a grant and as part of a very generous donation by a family who has donated to the museum. Yeah.
Is this capable of video?
Yes. Or? Yes. You can also take video. You can do 3D imaging on this. You can do high definition imaging, which high definition is really good when you really want to see morphological characters, but aesthetically is not as nice. So that's why I don't use it. But yeah, if you're taking images of something for a publication and you really want to see like all the nooks and crannies, and using high definition, actually using HDR mode is a really good option. So there's a lot of different capabilities. I mean, I'm just showing you like the most basic thing that I do when I do this 3D stacking and stitching. But this whole, you can actually take this whole setup. So the camera, the scope basically, and the lens, and the whole thing can be tilted. You could take it at all these different angles and just do incredible like 3D imaging. It's pretty elaborate. And this one is an older model, so they're up to I think like the 7,000 now. And that one, rather than having to manually change the lenses, it has all the different lenses attached, and it can turn around. It's
very good. Very nice.
Also, a quick question, Lisa. What is the range of magnification? Do you know?
So on this lens, this one is 20 to 200. But we have a lens that goes all the way up to 1,000. And then I think on this one, you can get lenses that go up to like 3,000.
Oh, wow.
Yeah, yeah,
Amazing.
yeah. It's amazing, exactly. So there's the finished product. I did goof right here. I don't know, you can't tell. I shouldn't have said anything. But yeah, it's a little, like a tiny bit out of focus here. So I would redo it. Usually when I'm doing this, I'm not chatting at the same time. But yeah, you could see it's, I mean, and this is actually doing the kind of just the auto, it's on like autopilot, but you can go in and you can change like the pitch, you can change like how many images it takes. So it could, if you really want it to be like super pristine, it can take a long time, but you know, depending on what you need it for. Typically, if I'm taking a photo for something that's going on Instagram, I'm just going to do it quick because again, like this is just something I do sort of, it's not like my main responsibility. It's just something I started doing because I enjoyed it, if that makes sense.
Does it tell you how many pictures it has used in order to stack?
Yes, actually, that is somewhere, but I don't remember where, but yes.
That's
yes.
okay. It was just a
Yeah.
question, but it tells you how many it's using or whatnot. So you can
Yes.
actually ask it to do more.
Yes, correct.
Oh.
Yeah, yeah, that's in the stitching setup here. One of these, I think it's in this one. Oh yeah, here. You can tell, you can set that amount.
Very good. Very nice. Very nice. Very good, Lisa. People have any questions for Lisa? Great job. It looks like a lot of fun, a lot of work.
Just a note that if anybody wants a really cheap version to be able to take auto montage photos in the field, there's a number of cameras out there that'll do this, and you can set the stacking from 10 to, I'm not sure what the top level is, but one of them is, I have it right here, the Olympus Tough Stylus. And that takes macro photography, really quick auto montage photos in the field, which is nice. But there's a number of those.
Very good. I know there's several stacking programs. I don't know what's out there anymore. There used to be one that came out of Russia that was very popular for a while. It was cheap, it did a good job, and can be worked. And they're on their second or third revision, they actually hooked it up so that it would take control of the camera, primarily the Canons, which is what they were using. And it would do the stacking and focusing and everything for you. But that's quite a few years ago, so I have no idea where they are right now. Oh,
Hey, Lisa, how do you clean your specimens when you get them out of the bug soup and prepare them for photography?
It depends on what it is. So with smaller size specimens, I use well, first we go through and we do several kind of baths, like sifting out all the ethanol and trying to get all the scales out of it. And then when I dry them, I use HMDS drying process. Or, with larger beetles and things, I just pin them and air dry them, or sometimes freeze. I mean, it just depends on what it is. If it's a bee, then of course I do the whole washing and blow drying and do the bee beauty salon process. And then from there I use, sometimes I'll use, no, I don't use pins that often because that's too dangerous, but I'll just use like a little brush that's just down to like a couple of hairs on it. So I've cut this brush, it's probably hard to see. And so I'll go and just kind of try to brush it out to get any dust or, any scales off of it.
Is that what you're doing with your parasitic wasps primarily?
It depends on how small they are, of course.
Okay.
Yeah, the really tiny ones that just kind of have to get lucky. And then from there, I don't know what a camel, no, it's just it's not a camel hairbrush, but I think I need to buy one. And then from there, I would just go through and clean it up digitally. So I would just, blow it up as much as I can and then go through and just try to get rid of any imperfections.
Right.
So I try to do as much as I can on the front end, and then anything that's too dangerous to do manually, I'll do digitally. Yeah, and Kim was asking me, because I don't clean up photos at work, because again, this isn't like my main job. So I'm at home in my jammies watching TV while I clean up photos. Yeah.
My
Hi,
sister,
Lisa.
my sister gave me this little microscope that plugs in with a USB. I haven't used it yet, but it says it's from China, and it says looks like it goes up to 1,000, which I'm skeptical of. There's a little way of adjusting the focus, but this must be fairly cheap, and it's made for computers. So I haven't tried it yet. There are LED lights around the lens. So
It has a very shallow range of focus. So it's a challenge to use with an insect, in my experience.
Depends on which model that you happen to get or who's you're using out there. BioQuip sells 3 different versions of a very similar item, or they did many, many years ago. The outfit is actually located in Torrance. And I've seen them. Art, have you seen their demonstrations at the ESAs? They can do some fantastic work. I've got 2 of their cameras. I've never been able to achieve what they have, but it depends. Their depth of field can sometimes be a little bit narrow.
Well, I have the exact unit that Steve has, so that's what my comment was about.
Okay, so. Just
Hi,
enough.
Lisa. I had a question. For your wasp that was a female parasitoid wasp, the one that had the ovipositor, what were the 2 other filaments that were hanging off?
That's actually a sheath. So when that wasp was alive, that those 2 parts of the sheath would have covered the ovipositor. That's a great question. I should have mentioned that. And after they're dried, those 2 part like lengths of the sheath will usually kind of get dried out, and sometimes they even kind of coil or pull away from the ovipositor.
Oh, okay, that makes sense. Thank
And
you.
I don't know if there's somebody here knows if there's a more technical term for that structure, but yeah.
Okay, thank you.
You're welcome.
Okay, thank you very much, Lisa. Do we have any more questions or?
Yeah, just real quick, I can follow this up later, but seems to me if we're getting back to doing live classes on campus, Lisa, maybe I should have our entomology class get a malaise trap and do it on campus somewhere that hopefully won't be, student abused or weather abused, and we could try to contribute to the bioscan project by having something right on campus at El Camino.
Oh yeah, reach out to me and we'll talk.
Sounds good.
All right. Anybody else have any questions? Can people bring in specific specimens and ask you to take pictures?
Well, I don't know if I want to open up that offer, but I don't know. I'll just say have people contact me privately and I can let you know if it's something that I can do.
Okie doke. All right, Lisa, thank you very much. We appreciate you sharing your knowledge and your time. Thank you very much. Okay. Jim, are you around?
Yes, I'm here.
Ken, before you go on to Jim, can I ask what you mean by stacking? What does Lisa mean by stacking?
Okay, yes, I'll answer that really quickly, and that's probably going to be talked about with the other 2 presenters too. But that means when you're taking the photo that has a really large depth of field, like especially something that's highly magnified, it's like you're taking it in slices,
Okay.
right? Because you're going up, focusing, and so there are programs that will then take all those images and combine them so that everything is in focus. So that's what you mean by stacking. And then stitching is the same process but side to side, because it's taking a part of it here, an image here, and then it's putting it all into one like composite image.
What was that word? I didn't catch.
Stacking and stitching. So
Dishing.
yeah.
Okay.
Stitching would be, in your XY, and then this is your stacking.
Okay. Thank you.
You're welcome.
Okay, anybody else? All right, then if we might welcome Jim.
Ready?
There we are. Dr. Hough, past president, longtime member, contributor, etc. to Lorquin. How long you been at Northridge?
25 years.
25 years. My goodness gracious.
Yeah, don't.
How many more are you looking forward to?
Until I'm dead, perhaps. But some people
Okay.
leave here feet first, and I hope to stop before then, but we'll see. It's a good job.
Yes, and as long as you enjoy it, that's a fantastic thing to do. Jim grew up in Los Angeles.
No, I
So-
can't. I'm at a Zoom meeting right now. But have you heard anything? Who's winning? Who's winning in the Astros and the? Hello?
Hey, Mike, you need to mute.
I heard Astros and
Mike, turn off your microphone.
Mute, Mike.
Okie doke. Anyway, Dr. Hough grew up in Los Angeles, got his BS in biology at I think that stands for UCLA. And after that, earned an MS and a PhD at Utah State University. How long were you in Utah?
And I accidentally muted Jim when trying to mute Mike. So can you take
Okay.
yourself off?
Well, I got
But
muted by mistake.
Yeah.
I hate to reveal how long it took to get my degrees. I'll only say that I went from kindergarten until I never missed a semester or quarter of school from kindergarten until I was 40.
Do you still have the certificates of attendance?
Uh-huh.
Okay. Anyway, we would like to welcome you very much. And you're going to give us a little bit of insight in using a camera in the field as opposed to a microscope.
Correct.
Thank you,
Okay.
sir. It's all yours.
All right. We go like this, this, slideshow. How's that? Can you see a picture?
Yes, sir.
Okay, so I take one kind of picture of insects, and this is the goal that I'm after. And I'm going to be giving you some information and some tips on taking these kind of pictures. I almost always strive to take these lateral shots to capture the essence of the whole insect. And I almost always try to get them facing left to get this kind of a product. The uses I have for these pictures, I'm trying to take pictures that I will use in publications, either my own or other people's, in talks that I give, such as this, and in lectures. So those are my goals. And it's a little bit like collecting insects that you put on pins, but this is collecting images of them. And this is the only one I'll show that has the name or information written on it, but that's an easy thing to do to make this an entity that can be useful for many ways. I've been doing this for a long time. This is a picture of me as a 13-year-old taking pictures of insects with a close-up camera. And in that time, in the three, almost 4 decades of taking pictures with slide film, I was able to take some good pictures. Okay, now this is not something that I figured out on my own or learned. I learned how to take pictures from my father, who was the curator of entomology there at the Natural History Museum. And he was taking pictures in the mid to late 60s using a setup that he made in the garage that is a single lens reflex camera with slow slide film and a homemade contraption for holding strobe lights. And the idea here is to have some light coming over your left shoulder that is kind of like the sun. And then the light on the right-hand side is used to get rid of the shadows that are created by the other light. So here's a picture of him taking a picture of some insects on some flowers. And here's the setup. It's just a camera with a bracket holding some lights. And this was a little bit cumbersome to use because every time you changed the focal length by dialing the lens or using a different number of extension tubes, which are these devices here that are just hollow tubes that move the lens farther away from the camera and hence increasing the magnification. Every time you change that, you had to change these pieces over here. As we'll soon see, the setup that I have now is superior to this, and there's not a lot of fooling around. But with trial and error, this was a real good system, and it really worked. Then you couldn't get the film, and you could hardly get it developed. So I went into a funk for a few years and got all depressed. And then I saw the future, and the future is in digital photography. One of the best things is you can see your product right there. You don't have to wait 2 weeks for it to come back in the mail and to realize that your picture is good or your picture is no good. And so what I did was I recreated this slide film setup with a digital film setup. So it has a camera base here, extension tubes, a macro lens, and these two strobe lights. It does the same thing. And I can remember the delight I had the first time I put this together and put a bostrichid beetle in front of me to try and take its picture. And it was great. And it just opens up a whole new avenues for taking pictures. And so from here on out in this little presentation, you'll see pictures that I've taken with this setup, and we'll talk about how to do that. I put together a picture of the parts. Should you be interested in this? Now you see the total estimated cost up there. I just found these same parts that I had on Amazon. This is just a hair cheaper than Lisa's camera. And these parts, probably some of them, like the body up there, when I bought this new, it cost a lot more than that. So if it was me, I would be buying a more up-to-date camera body. But these parts, it's not too bad. If you're really interested in taking these kinds of pictures, it's really not too prohibitive. And so I've got them all listed here, and especially since this is being recorded, somebody could go back to this and look up these parts to recreate the setup that I use to take the kind of pictures that I am interested in. As far as taking pictures, I hardly do this anymore. This is Art Evans wallowing around in the desert. And man, I can get down there. It's like getting up is the hard part. And a lot of what I'm after in pictures, I really can't do it very often by taking pictures in the wild. Other people, this is what they do. I've pretty much given this up and take pictures in a different way. Also, it's real hard to have your camera and your photography stuff and your insect collecting stuff and manage that at the same time. So what I tend to do now is I collect insects and then I take them back and take pictures of them later. So here's a setup I did day before yesterday here in my lab. And it's pieces of wood and rock and plants and flowers, all kinds of things. Because insects are small, you can build a little semi-natural setup and release or put your insects on these things. And nobody else knows what's around you because those don't show up in the pictures. And so you can take pictures that are that look natural in a controlled environment. And so here's a little setup here that I did day before yesterday to illustrate this. And then here's a picture of me taking pictures of insects. Okay, so it looks, hey, that's pretty simple. Do these things look any good? I think they do. Actually, when I picked these leaves of this poplar, I didn't notice it at the first, but there was a spider on these leaves, and that was this jumping spider here, Phidippus audex. So using this kind of strategy, I take pictures of insects that are, again, this is the whole thing. To me, this is capturing the essence of the insect. And like I like it, it's facing to the left. And here's one that's sitting on its own background. And so this is fairly easy to do. You can point both of your lights at the insect and it illuminates both the insect and the background. You can also take pictures where the insect has space behind it. So a lot of times they're not sitting on their own background like this blister beetle. There is stuff behind it. So you want to put objects that are kind of out of focus behind your subjects. And sometimes you can take the pictures and that turns out to be black. And you get a picture of an insect with a black background, which in my book is not very preferred. But you can take the light on the right and you can move it and point it toward the background and it will illuminate the background like this and look much better. So here's an example of this blister beetle. And here's an example of a wasp using the same thing. Again, I'm collecting these things live and I'm turning them loose in a somewhat controlled environment and letting them do their thing and being patient and trying to take pictures that meet my goal, which is what we have here. You can also take pictures of parts of things like Lisa does with their heads and their faces and capture a little bit of a different essence rather than the whole insect, like this local katydid here or this praying mantis. You can also take pictures of things that live in water. Those were all terrestrial insects. To take pictures of things that are aquatic, I have this is a really small aquarium. It's only about 3 inches square. And I have a little piece of glass in there. And what you do is you take this, fill it with water, put some substrate in there, and then turn loose the subject for your photographs, like something on the surface of the water, this whirligig beetle here. Or you can take pictures of things underneath the water. So this is called Drunella flavellinea. This is a mayfly larva. And this mayfly larva is in this little teeny aquarium. And you sit there and wait for them to get into a position, or you prod them to get into a position that you like to take the picture. It helps to learn a little bit about insect behavior to do this, and all of them behave in different ways to different stimuli. You can have something running across a substrate and you lightly blow on it and it will freeze. I lightly blew on that jumping spider and it went berserk. So don't blow on the jumping spiders anymore. You do by trial and error and figure out what works for them. That jumping spider, any quick movement, it saw that and was very active. But once I got it to settle down a little bit, you could approach it very slowly, and with shadows and a stick, you could get it to walk one way and walk another. So it's kind of fun sometimes to figure out ways to get these insects to do something so that you can get the final product that you want. Now, let me give you some tips that I've learned. And for some of these pictures, I'm kind of particular about stuff that you might not think about. And so I want to go through a few of those. A lot of times people keep insects in the refrigerator. Maybe you want to keep them alive for several days. You can't get to them for a while. And sometimes people use cooling them down as a way to remove, slow down their activity. This is what it looks like when you take a picture of an insect that's cold. It looks just like you. If I put you in the freezer for 4 or 5 hours and took you out, took your picture, it's not going to be a very good picture. Okay, so here is a a bibionid, Bibio albipennis, and it looks awful. So you can make a good picture of something, but if the posture and its pose are no good, I think it's a terrible picture. So if you do cool down things or do other things to them to slow them down, give them a chance to completely warm up and become active and get a more natural posture, or don't cool them down in the first place. So here's this fly that I think looks awful because it's been cold. And here's the same fly before it was cooled down, or after it was cooled down, but given plenty of time for it to warm up and move around and get a more natural pose. Okay, here's another, this is a picture of a canopid fly, and you go, hey, that's a pretty good picture. You did a good job, but I don't think this is a very good picture, okay? Here's something else that's being lazy. You might get this also if you cool this down. Yeah, it's standing on its 6 tarsi, but it's also sitting down on its belly, okay? They don't normally act like this. And so that's not normal posture for these flies. So not cooled down and or given a chance to warm up, this is how these flies stand. And so for your picture, this is more about what you're after. You can take pictures of insects that are in focus and the color balance and everything is good, but I really like putting some effort in getting the postures right and making them look good. Here's another example of a problem. Now, yeah, okay, I don't like this one because it's facing to the right, but that's not that big a deal. But look at the background here over the prothorax and the top of the head. It matches the color of the insect. And so the boundary between the insect and what's not insect is obscured. It's not very clear. So pay attention to your backgrounds. You can manipulate these things when you're taking these staged photos. So here's the same beetle where the front part of its head, which is dark, has a light background. And now I can see this much more clearly. So it might be something you might not pay much attention to while you're taking the picture, but you'll notice it later if you're kind of picky. Here's another example of the background. This is Idiostatus inermis. It's a local katydid, but look, I've lost most of the antennae. You don't see them very well. And the ovipositor almost matches the wood of this background. So it's not very clear. Give it a chance to move around and encounter a different background. This is not the best for the antennae, but look at the ovipositor now. It's so much clearer and distinct as being a part of this insect. Lots of, forget that this thing is missing a leg. A friend of mine that went to Australia brought it back and it was still alive. So, hey, I'm gonna take a picture of it. So these, you might say, hey, this is a good picture of that beetle. It is, but it's kind of like it's asleep. Okay, what's missing are the antennae. And so this may be a good picture of a beetle, but this is better. Okay, so give things a chance, again, to get active and to move around. And lots of beetles tend to do this with their antennae. They'll hold them under when they're, I don't know if you want to use the word scared, but they'll hold their head down and their head under. And then if you give them time, they will kind of come out and start looking around until you move around. But give them a chance to do that, and it will make a much better picture. Even something as simple as having a curled under tarsus, okay? So that's okay, but that's better. Okay, so this is another reason for which you can do well in digital photography. Take a bunch of pictures. I still take one, then another, and I wait. And though that goes back to the old slide film days when each one of those cost $0.25. And so take more pictures. You can throw them all away except the 2 or 3 that you really like. Okay, here's some other things to pay attention to. Here's a picture of a beetle. The head is all out of focus. If you get any part in focus, really try to get the anterior or the head end of the insect in focus. The pictures that I take, if you take any pictures of any decent sized insects, there are parts of them that are out of focus. There's no stacking, there's no stitching involved. That's what Lisa's technique really does. And you see that in those photos. They are clear front to back, top to bottom, but you can still make your pictures better. So here's a picture of this beetle with its head out of focus. Here's a picture of the same beetle a second later with its head in focus. Okay, that's a better picture. Sometimes you don't solve your mistakes at the picture taking phase. You can solve your some of your problems later on. So here's a picture of this local longhorn beetle. It's kind of dark. You can work on it in Photoshop. Photoshop and can be intimidating. But if you learn about 6 easy to learn things, you can really manipulate your photos and make them better just by manipulating the shadows and the highlights. And with a little bit of work, Lisa talked about dust and dirt. This happens sometimes when you collect things, keep them in a vial and take stage photos. They can get dirt on them. If you don't remove it before you take the picture, yes, it takes time, but you can remove those kinds of things in Photoshop with just a couple of minutes of training. It's not too bad. The other thing I want to talk about is, what do you do with all of these pictures that you generate? How do you manage them? Okay, so here is a photograph of Seriana tridens. It's a local wasp mimic syrphid. So I have this picture and I have lots of them. So I use a program that's called Photo Mechanic. There are other programs that do similar things. You can see in the back, here's a bunch of my syrphid pictures, kind of like on a slide sorter. And for each of those slides, it has a page here. And here's that same photograph. And I fill in these fields just like I would on an insect specimen. So up here is all of the label information. This is exactly as what I would put on the pin of this insect. This is what the object is. Here's me, the photographer, and then I give them a catalog number. All of these fields are searchable and retrievable. So you can ask of your program of all your images, give me all of the syrphidae that I took in Kentucky in 2015. And instantaneously they show up on this slide sorter, but you have to put in the work, and it takes a long time to put in your work to organize your images, label them properly, and put this kind of data in. Another big plus for taking pictures of things that you collect is that I can retain the specimen that's associated with the image. And so if it's not something, and oftentimes it is the case that you can't identify something based on its image, well, I have the specimen. And so what I do, here's that same picture. I still have this specimen. Here it is on the pin, and here are its labels. Our locality label, like we put on all of our specimens. This is a catalog number for this specimen. Here is the determination label for that specimen. And then here is my photographs. So if I see this specimen, I can go retrieve my photographs using these catalog numbers, and vice versa. If there's some question, if I showed a picture to somebody or somebody wanted to use it in a publication and they said, I don't think that's right, I can use these numbers and go back to my specimens and look at it again and see if a mistake was made. Okay, so that's the kind of pictures that I take. If that interests you, it's not too expensive to put together a rig that can take these kinds of pictures with a handheld camera. And then some tips on making better pictures. You can always make better pictures. and then spend the time and get your images organized. That makes them a lot more useful to you and to other people. Okay.
Very good, Jim. Thank you. Have you ever used a reversing lens?
No, I'm kind of, once I learned one way and it made a picture that I like, I quit learning.
Just a question.
I'm done. Yeah. No, I haven't never used that, but I've heard of those.
Yeah. All right. Very nice. Very good. You prefer the Nikon?
Yes, and I don't know why I chose that. I think my other camera might have been Nikon. I just walked into the camera shop and I said, do you have this? Do you have this that matched what I had before? And then I took it and just in 10 minutes of fooling around with it. But these cameras now, they're so sophisticated, but I only use 2 of the features because that's what I use it for.
Very good. Are you using Photoshop's cloud version?
I don't think so. I just use a regular, our school has a site license for the Adobe things.
Okay.
I've tried Adobe Illustrator before too. If you're intimidated by Photoshop, don't touch
go there.
Illustrator, man. It's very sophisticated, but I learned just again about a half a dozen things on Photoshop, and you can do wonders. Yeah.
Very nice.
Hey, Jim.
Yes.
Do you prefer to take pictures of insects that are facing left, and why?
I always do this with insects, but bird specimens, mammal specimens, everything. I treat the left side. The left side is the sacred side. Don't fool with that. So you tie tags on the right foot. You put the pin through the right side. I always leave the left side alone. If I'm making a mammal specimen and I want as many bones as possible, I don't wreck the left side. So I leave that. So I think that's why I like the left side. And you put stuff on points, insects on points, you glue the points to the right side. You kind of mess up that side. So I like looking at the left side.
Jim, I remember your dad telling me early on that the insect always had to be facing left, and there was no ifs, ands, or buts. That's just the way it was. And I suspect he had a greater influence on you too.
It probably, once you start something like that, and then I do something today because that's how I did it yesterday.
Yeah.
And so it very well could be like that. And sometimes just to get a picture, if something's really active and you're having trouble and you get a picture and it's facing right, go ahead and take the picture so that you have it. And then somebody that's good at Photoshop say, hey, I can take that picture where it's facing right. I can flip it horizontally and now it's facing left. Why should you not do that? Lots of insects, they're not always symmetrical.
Right.
Yeah. So don't do that. Yeah. Yes.
Yeah. The photo mechanic software. Did you try other software?
No. I just asked a couple of other people that what do you use to manage your photos? And Riley Nelson at Brigham Young, he said, I use this. So I said, okay.
Probably what I'll do.
The thing about that is it's really made for newspaper photographers, and it has all these fields about things that you would never use. And I don't know how to, I would like to get rid of them and only keep the ones I use. But it has all kinds of information that a person that took a photograph that they might use on a news broadcast or in a newspaper. So photojournalists, that's what I think it's geared for.
For people that are using Apple computers, you've got iPhoto that's built right in, and it's very similar. And I'm like you, I'm not a tinkerer. I just want to do the handful of things that get me where I want to go. And iPhoto is very intuitive. I realize not everybody uses Apple products, but if you have an Apple, chances are you've already got something there that would be very easy to use and certainly to get started.
Lisa, you have a question.
Yeah, a quick comment about Photoshop if people are curious. There's a version, like a scaled down version called Photoshop Elements that's pretty affordable. It's $100, you pay once. And it has a lot of kind of quick guides that will sort of do the work for you, but also kind of teach you how to use it. So if anyone's curious, you could do a free trial and then check it out. And then Jim, I had a quick question about the aquatic setup. It looked like you had like a glass slide over the top. Did I see that correctly?
You saw it incorrectly. I just broke the thing, and so I spent day before yesterday, gluing it together and redoing it. But it has a glass front, and then there were two more glass panes, and so what you could do is you could turn something loose, but with a piece of glass, keep it toward the front so it doesn't go to the back.
Got it.
So what I'll do is put something in there, have that piece of glass, and you could put leaves and stuff behind the glass and you see right through it. The thing about using that and taking pictures, you got to pay attention. Bubbles, there's lots of bubbles. So you should spend some time to get rid of the bubbles before you take your pictures. And I'm thinking of some ways to get rid of these, but there are straight lines in there too that you see sometimes. Some of that you can remove later, but it'd be better to take care of that stuff ahead of time.
That's a technique that's used in doing tropical fish. A lot of tropical fish, people who take them will put a piece of glass in back, then push the fish towards the front so that they get a better picture, more or less.
Yeah. Otherwise, this thing's going to go to the back and it's going to hide. You can't do anything except sit there and look at it, poke it with a stick, and hopefully it goes to the front. Yeah.
So
Okay.
Jim, how do you keep things from flying away? Like you've got wasps. And
I never said
flies
I
on a piece
did.
of bark?
I never said I did keep them from flying away. You know, one thing that's worked very well is sometimes on trips in the tropics, the beds have mosquito netting over them. And I've spent so much time underneath those. It's great because something flies away, it's in the mosquito netting. The only thing is it's in those parts of the world, it's hot and it's humid, and that mosquito netting really slows down the airflow. So you sweat like crazy under those things. But I'll taken pictures in my camper, and things get away, but you got to try and catch them fast and don't give them a chance to get away. I take pictures here at the school, and I get most of them back, but the lights are open, and if they go up in the lights, which they tend to do, that go toward the light, yeah, they can get away. And that's real frustrating because now you took a picture of something and it's gone. So yeah, things do fly away. I spend a lot of time with something loose, and all you have to do is put sometimes a vial over it, let it walk for a while and pay attention to it. Some things will behave and other things won't behave. And it could be so frustrating, and then you just smash them and then I win, but you didn't get a picture.
Jim, when I was working with spiders, I used a lot of CO2, and that works well for anesthetizing them if you don't over anesthetize them. And then with spiders, it doesn't necessarily work with insects, but spiders can't fly. So I let them go on a carpet and just chase them around the carpet for a few minutes, and they tire out. Spiders, this works really well because they don't have an efficient breathing system, and they tire out. And then you have a few minutes where you can take lots of pictures of them before they wake up. The only problem was the brown recluses because you anesthetize them or tire them out, and they'll just sit there quietly, and then they go ping and run. So you just don't do that with a brown recluse, and you're fine.
Yeah, each thing is its own little issue, and which is kind of fun because you spend time with the insect. Usually we just see something, catch it, kill it, deal with it later, but we could spend time with them when they're alive, and you learn stuff, and it's a great experience. And taking pictures, you get to do that, and you get to see something. Each of those requires a different solution to get them to behave. Some things are just great, and other things, they can be so frustrating because they won't stop moving.
Jim,
Yeah.
A couple of comments. One is I've learned, like you, and it was good points, that when you're taking a photo of an insect, you're so focused on that insect, you forget about the background. So I keep that in my mind, the insect, the background. So I don't forget about the background. And then in terms of cold, because I've filmed commercials and TV shows, and I have an American cockroach, and I use cold all the time. I'll use CO2, I'll use cold. But what you have to learn is the behavior it goes through. So the roach, and I'm sure all insects go through certain behaviors. So when you first put them down, they look dead, they look, so you want to see the behavior. And one of the things they'll often do is clean themselves. And so I've watched that cleaning behavior. You can see this with ants. And so when they start to wake up, their behavior is to clean themselves. And in regards to flying off, because of my bug art painting, I discovered this, because people ask me, how can you make bees walk around? How can you make flies walk around? Don't they fly off? Well, one of the behaviors, you won't do this, is to put paint on their feet. It's like mud. An insect isn't going to fly off with all that extra weight. The first thing they'll want to do is clean themselves and then fly off. And so I get the painting done with all the weight on their feet before they fly off. So I know you wouldn't use that, but I just wanted to.
Yeah, but it's a good example of figuring out a solution to the problem. That can be fun. Yeah, and then when you get lucky and you get a nice picture, then it's rewarding.
Steve, how do they clean the paint off of their feet?
I do that. That's my job.
Okay, Jim, thank you very much. It's very enlightening. Lots of good pieces of information, and we thank you for sharing very much.
You're welcome.
Okay, next up, Kim, are you still here?
I am still here.
There you go. All right, Kim Moore is our next presenter. Anybody have any questions for Jim before we move on here? Okay, Kim Moore left apparently a burgeoning career in information technology, obviously quite good at it, and pursued interests in nature. She has taken several classes in entomology, zoology, birding, botanical illustrations. She does some great pictures, incidentally. And she's a certified naturalist through the Los Angeles Coastal California Master Naturalist Program. So besides being an insect person, she's also a bird person. And I believe do you have a regular meeting at the park?
Not at the park, but I do have a regular Zoom meetings for birding and then one for insects.
Okay, very good. Anyway, with that, Kim uses a phone for her camera. And with that, I'm going to turn it over to her.
Okay.
What kind of phone?
Well, I don't use a phone. So,
okay.
I use a setup similar to Jim's, but we'll go through it.
Okay.
Okay. I'm going to share my screen here. Okay. Yeah, I do occasionally use my phone. I shoot, as Jerry said, I'm going to close up this thing on my side here. Okay. As Jerry said, I shoot a lot of wildlife. I shoot birds and insects and actually anything in Southern California. So lizards and snakes and anything I can focus on. But I do particularly like birds and insects. I started with insects and then moved over to birds. And I don't consider myself an expert in anything, including photography and entomology. But I do get some good shots that I am happy with. And some people do request my shots and I use them in presentations. So it's really what your purpose is of why you're doing the photography. I'm doing it for my own purposes mostly. And I'm going to show you my equipment and the settings I use on some of these. And not knowing everybody's interest level here, some of it I may give a few technical details, and then at the end I'm going to show my some images so you can bear with me. These are my cameras. I've had other cameras in the past. I use my Olympus D7100, so that's very similar to Jim's, as my primary camera, and that can I swap out with a lot of different lenses. You can use a cell phone. I have used my cell phone. I have the Olympus TG5, and somebody else showed that here earlier. I don't get as good pictures with those, but I stick those in my pockets. So I do use those. And I also have a whole bunch of different lenses. I use macro lenses and I use zoom lenses. Obviously, you can see my big zoom lens for birds, and then I have my macro lenses for insects. But I always say the best camera is the one that's in my hand. I'm an opportunistic photographer, and I like to be out in the field. So whatever I'm shooting is something that I will use for what I'm shooting. And you can have, so if you have a different lens, I mean, you may have a Canon, you may have a cell phone, you may have whatever kind of lens and whatever kind of camera you have, you can get good photos of insects if you know how to use that particular camera to the best of its advantages. So it really depends on what you want to shoot. I usually take the camera with me for the type of outing I'm going on. Sometimes I go out with people who are interested in insects or black lighting, and I'll take my macro setup. But if I'm out with birds, or I'm going to go shoot dragonflies in flight, then I'll take my long lens and a different camera. But I will shoot with whatever I have. I have had my macro lens on shooting insects and then come upon a rattlesnake or a bobcat. And I really wish I had my long lenses for that. But if the macro lens is what I have on my camera, I've actually gotten shots of rattlesnakes and bobcats with my macro lens. And similarly, the other day, actually, I was out birding, and I came upon this really unusual spittlebug. And so I used my zoom lens, a 600 millimeter zoom lens, to take a picture of a spittlebug. So I just say, take what you've got, but learn how to use it. So this is my insect camera. I actually have two cameras. I have two D7100 cameras. So I have this rig always ready. And then I have my other one on my zoom lens, which I always have that one ready. And it's nice to have the two, so I don't have to be changing lenses between my purposes. And then I keep my insect camera just pretty much all the way set up, because whenever I leave the house, inevitably there's an insect that's on the front wall that was left over from the porch light the night before, and then I have to run back in and grab my camera and take a picture of it. So I have it already set up. I have a Tamron 90 millimeter macro lens, and then I use a speed light. And the reason why I use the speed light is it's a really good quality light. It's good for a number of purposes, and somebody gave it to me. So it didn't cost me anything, and it's good quality. But you don't want a harsh light on your insects, so I use that diffuser, and it's an inexpensive softbox diffuser to go with my flash. And so that's what I have in the setup. I have the flash on the camera, and then I have the diffuser. I have it like this. I saw Jim has them on the sides. On the sides is probably better, but this is the one I have, and this works for me. I think the other thing is what settings you have. I do manual setup on that, and I just have pretty much the same settings. I do change them a little bit. Actually, I use ISO 400, 1/250th, and f14, and do manual focus with it. This is really good for insects or anything that stands still. But of course, I like to shoot things that move, and so I do it. It's more challenging with this setup. A brief thing on aperture. We've talked about depth of field. Lisa does the stacking, and Jim was talking about getting the eyes in focus and trying to get depth of field. The smaller the aperture, the better the depth of field. So the f/16 is a small aperture, but you need more light because now you have this small hole. So that's why I use that light system to add more light to the subject. This is the lens I use the most often these days because I'm shooting more birds than insects. But I do use this for insects a fair amount, and especially because I like to get flying insects and the interrelationship between insects and birds. And this is a zoom lens. This is 150 to 600 lens. I also had an 18 to 400, which is smaller and lighter. You can see this is pretty big and unwieldy, but that's what I use and I like it. It's heavy, yes, it is. This is, I use it for the birds and the insects, but also dragonflies and damselflies, dragonflies and butterflies in flight, you really can't get with a macro lens. They're never going to sit still. And the distance, and you can get better distance to the subject with this. My normal settings for this one, I use aperture mode so that the camera determines what the shutter speed is, but I adjust the shutter speed by changing the ISO of the camera. And so I'm always looking at what is my shutter speed, and I'm trying to have a relatively quick shutter duration, but then that's just a rule of thumb. And then if I'm going to shoot an insect like my spittlebug with a long lens, you have to stand a long ways back from it. So that's a bit of a challenge. So I wouldn't necessarily use this for that. Because of the distance, I'm using a shallow depth of field here, but I like to blur out the background. So I have a wider aperture and therefore I don't need the flash, but then using a fast shutter duration will freeze that image. Focusing, my camera will allow up to 51 focus points for the camera to automatically focus. That's ridiculous. I only use 2 settings. Like Jim, I have my settings. The camera is capable of so much more, but I only use a few. I use single point focus for my macro, and if I'm shooting anything still. And then if I'm trying to shoot something in flight, whether it's a dragonfly or a bird, I'll use this 9 point. So anything that hits that 9 point will automatically focus on. And I use continuous focus when I'm using something in flight. And you may wonder what some of that is. And for that, the best thing to do is read your manual, your user manual, to figure out how to do the settings. Look up continuous versus single focus, and look at how to change your center spot focus, focusing and look in your manual. And your camera, whatever it is going to have something equivalent. And so you just want to get to know your equipment the best. Lost my place. That's it. So here are some of my shots. And what I like to shoot is in the wild, different behaviors. This is a tropical house cricket that was being eaten by a brown widow. And then this little male came in. And so he's sitting there kind of on the edge while she's eating the cricket. For a while, I don't know where they all went, but I had a really lovely collection of green lynx spiders in my front yard. So on the left, which I shot with my macro, these are mostly my macro shots. I had this spider that had captured a bee, but then I had all of these little flies. Oh, I was going to lay below those. These are those freeloader flies that are coming and eating the bee and annoying the spider while the spider was eating the fly. And I did work to try to get a neutral background, like Jim was talking about. And then on the right was 2 egg cases that this spider, and you can see that she's grabbing, actually she's spinning the egg case on the lower one, a second egg case, while her first egg case has all the spiderlings coming out. This one I had my macro. I think Doug Copeland, you and Mike Martinez, you were with me when we were at El Dorado Park, the nature center, when I shot this. And this was a bumblebee robber fly that was, I just noticed, was laying its eggs on this stick. And certainly that's something I have not seen before. And so there I took the shot with my macro. I wished at this one I had a longer lens because it was a little further away. But then I did a close-up over here, and the way you get a close-up is by cropping. And so I do a lot of cropping on my images. Leaf cutter bee, I did use the flash because I was using my macro. So this is not all the same. This is not like one continuous shot. I sat and I watched the bee, and the bee kept coming back to my rose petals. And so I kept shooting this bee as I was doing that. Again, trying to get the neutral background, using the best shots that I could. This was a syrphid fly that were mating, and they had just taken off, so I could use my flash and get them actually mating in flight. That was fun. This is the stuff that I enjoy, and I really appreciate that there are other people here who can enjoy this stuff, because I have so many friends that I tell them, oh, I've got syrphid flies mating in flight, and they don't have any enthusiasm for it. I don't know. This now is when I have to use my zoom. I have to go to my zoom lens. So this is with a 600, well, I guess this was at 550 millimeters of my lens, but to get these butterflies that were cavorting. I don't know whether they were territoriality or they were doing some courtship, but to be able to get them in flight. Dragonflies also in flight, where he's about to attach to the female. And this one was what I have is my Nikkor 70 to 300. So this was a much smaller lens. This was before I got my 600 millimeter lens. But any kind of zoom lens is good for insect photography. And I think that people think insect photography, you think macro. Certainly, if there's a body of water between you and the insects, you can't get close without a long lens. And then, because I like birds, birds often find the insects for me, and other animals find insects for me. So here it's got a Jerusalem cricket. This was at the top of a tree. And I have now seen several downy woodpeckers that love these manted egg cases. And so I've seen this behavior a number of times. So I'm realizing that the tops of trees have a lot, especially at this time of year, start to have egg cases and larvae up in the top. And so when we trim our trees, you're trimming off the top of it, you're trimming off all this food for the birds and all these insects. And of course, Lisa, oops, that went too fast. Lizards also, I don't know, this, it won't stick up. I had a problem with my thing on that one. But as you can see, lizards also, you just have to be there to catch the moment. And so this one was my back tree. My fig tree has died, but has now become a home for carpenter bees. And as I was sitting, having a cup of coffee in my backyard, I saw this lizard come up, and I saw that he was eating the carpenter bees. So again, I had to run inside, grab my, no, I think I may have even had it next to me, my long lens to take a quick shot. So some of these things you just have to catch very quickly. And then I do catalog my things. I use Lightroom. I don't use Photoshop. Photoshop's more complicated for me. Lightroom is very easy. And I do all my cataloging in Lightroom, and also I can lighten an image. I can crop my image. I can sharpen the image. And lately I have found this other software called the Topaz products that have plugins to Lightroom that have really excellent enlarging and also sharpening capability. And then I use them on my own website. I post some on my Facebook page, and then also iNaturalist. I post a lot to iNaturalist. And so sometimes I shoot first and I get a crappy shot, and I'll even put the crappy shots on iNaturalist and then save some of the better ones for my own website. So it's really a matter of how much time you want to spend doing these things. Prepping and cleaning your cameras. Looking for insects is where I want to spend all my time in the field. Insects and birds and wildlife. Setting up scenes, which I think Jim probably spends a lot more time on that, and I don't spend much time on that at all. Photographing them is only a small portion of time, and then categorizing and labeling and editing them and posting them takes a longer amount of time. So if you're considering getting into insect photography, it's more than just the photographing them. And I didn't know how long I was going to have for this talk. I didn't think I was going to be here at first, so I created some shorter videos. Each of these videos is about 10 to 15 minutes long to talk about what I do for photographing insects. So I'm going to stick that in the chat so you can copy it out of the chat if you're interested, and watch those videos at your own time. One I did specifically on dragonflies, and then I did a couple on insects. Just the general insects. Part one is just, you know, thinking about what you're doing with it, and part two is the gear and the equipment, and then third is techniques. And so some of that may apply to you in what you do. And that's it. So I think we're on to the question part, if anybody has questions.
Very good. Thank you, Kim. Very, very enlightening. Lots of information. Yes. All right. Questions?
Thank you, Lisa.
Kim, on your dragonfly shot where the black saddlebags is about to attach to the female, how did you get such great lighting on that without a flash?
Lighting is, you know, I try to set my camera so that I'm going to get the maximum lighting on it. And then I will angle myself when I'm taking, you know, if you take photos and you're using something that's backlit, it's not going to come out anyway. It's like birding. When you see a bird up in the tree, I know Bettina's a birder, so, you know, and you got your binoculars. So you just angle yourself to get better light. And then also you lighten you can always lighten in Lightroom.
Yeah, I guess you know me and my photographs, I just point and shoot and hope for the best.
Well, that is what a lot I do. I delete a lot of photos and I don't show you those.
Well, I just noticed, I don't remember where I was shooting the other day. Oh, I know, I was at Laguna Coast Wilderness Park with Laura, and we were shooting a mantid, and it was a bright day, but the sun was already starting to go low in the sky, and our photos were all way too bright. And so, you know, I couldn't adjust for that anymore once I got the photos. But anyway, so I had the sun at my back and all the right things, but I just realized that my photos were too bright.
Then that must be the setting on your camera. You have to learn to set it for lighting. We'll go out sometime.
Thank you, Maze. Okay, any more questions? All right, we've taken more than 15 minutes a shot for each of the three of you. Time well spent. And I want to say thank you very much for great presentations, lots and lots and lots of information. It'll take us a while to absorb some of this. So without any ado, if anybody else has questions, please come in. Other than that, I'm going to sign off, go fix some dinner, and say thank you very much. Really appreciated. Jim, Kim, and Lisa, if you're still there. Okay, so have a great evening. All right, enjoy. Thank you.
Okay.
Nice to see everyone.
You
Yeah.
too.